Let's get started. If I cannot. Okay, so last time we were starting to look into the early years of microbiology field, part of the history, and then we said that we had the cell theory, which tells us that all living things come from living cells. And then something that also we need to understand is that back in the days, people weren't so familiar with how things started because it was early in basically well developed science. But we had some good scientists back then that they were thinking about, okay, why things are, including where life's coming from. So many believe in the 1700s that living things come from, or life arise from non-living matter, and that they called a spontaneous generation. But why did they came with this idea? Well, they saw, for instance, that out of decayed matter, maggots arise and then flies. And some people back in the day, they were puzzling why this is happening, and then many believe that, yeah, if you have a corpse, then you will have a living thing coming. But many they disbelieve that, and they start to prove if that was true. One of these was Francesco Redi. He was an Italian physician. And then to prove that that spontaneous generation theory was wrong, he basically boiled meat, and then he had the meat in a flask, and then he covered this meat with the lid. And the lid, of course, didn't let any flies that were around to go and lay their eggs, so that the eggs didn't develop into larvae, and then they didn't develop into adult flies. So he said, well, I mean, this is what I did, and no living things arise. So this is basically wrong, what you're thinking. So, well, that was his experiment, but many start doubting. Well, we need air to be present for these organisms to arise. So what he did then, he was same experiment. He boiled meat. He put that boiled meat into a flask, and it had a little bit of broth in it. But instead of covering it with the lid, that it was completely sealed, that flask with the lid, he put a gauze in the opening of the jar. And then, okay, flies were around, but the eggs, if they landed, of course, in the gauze, they didn't make it to the broth and into the meat. So then, no flies arise. And then they were saying, well, that can be true, but what about small microorganisms? And by then, they already know that microorganisms existed, because if you remember Van Leeuwenhoek, that merchant from the Netherlands, he already proved with his microscope that microbes exist, small, what he called animalcules existed. Excuse me. So basically what happened is that they say, well, what about small microorganisms? Where did they arise from? And then they started to do some experiments to prove where does these microorganisms are coming from. And one of these researchers was Louis Pasteur, was a microbiologist from France, and he pretty much did similar experiment, like he boiled chicken and corn, in a flask, a round flask with a neck. Let me go to the next one. But basically, the hypothesis that living things or living cells arise from pre-existing cells is biogenesis. So then Louis Pasteur, he did, it was boil this broth, and then he basically sealed the flask. And after sealing the flask didn't have any microorganisms. And same deal, there was someone asking, well, you need to have air. And since he was hypothesizing in his mind that microorganisms are coming from the air, what he did was actually to extend the neck of that flask, which was vertical, extend it with heat, and then create an S-shaped loop into which the ending of that flask was open, and air still can come into that broth. But the microorganisms didn't make it through this S-shaped loop, and they were trapped somewhere within that curvature, and that basically prevented microorganisms from growing. So there was a debate for almost a century about this, but it was until Louis Pasteur did these experiments into which he basically disproved this spontaneous generation. So then everything was put back into place just because there was this researcher, or well, it was going back and forth between spontaneous generation and biogenesis because John Needham put boiled nutrient broth into covered flask, and then when he didn't seal the flask, microbial growth occurred. But these flasks, of course, they have vertical opening, and that allowed the microorganisms to come in and then contaminate the flask. Many of these things nowadays we take it for granted because we are aware that microorganisms are found in air. However, back in the days was very hot topic. So then Lazarus Spallanzani in 1765 sealed the flask and then heat these broths to prove that John Needham was wrong. So 20 years after he did this, but again, they were like the opponents. Well, you need air. And then that's when Louis Pasteur proved that to be wrong. So in 1858, Rudolf Virchow stated that living cells arise from pre-existing cells. So that's the end of this era. And then again, here is the table showing you what Louis Pasteur did and then how he proved that spontaneous generation was wrong. So here is the overview of this experiment with these figures. So he poured this broth either from chicken broth or corn broth into this flask that has this short neck with this long part, and that long part was vertical, and he heated up, he make it as an S shape, and then he basically had this broth heated again, and then the ending of this flask was open, of this long neck, and then microorganisms couldn't make it to contaminate the flask. And he did many experiments, including experiments that basically helped to preserve beer and wine back in the day. Beer and wine was made sour by microorganisms that converted alcohol into acetic acid, and that was bad for people who made those products. So then he invented pasteurization, which basically is heating up liquids into a high temperature for a short time and then cooling it off to kill any spoilage microorganisms. So, what evidence supported spontaneous generations? Spontaneous generation, sorry. Maggots crawling in a decayed matter. And how was the spontaneous generation disproved? And what else? So sealing the flask is, yeah, making a long neck in a Erlenmeyer flask. Well, in that flask, it's not an Erlenmeyer, but very good. Okay, so now we're going to see the next phase of microbiology history, which includes explaining how Pasteur's work influenced Lister's and Koch's work, and then identify the importance of Koch postulates, and identify the importance of Jenner's work, and the contribution of Ehrlich and Fleming to microbiology. So the first golden age of microbiology is from 1857 to 1914. And this started with developing the relationship between microbes and disease, the role of immunity in preventing disease, and studying the chemical activities of microorganisms, having better microscopes and methods to culture microorganisms, developing first vaccines, aseptic technique, and the first use of chemotherapeutic drugs to treat infections. So, there was this researcher during that time, Robert Koch, which he developed Koch postulates. He was able to link the relationship between a disease that affects cattle and sheep in Europe that is known as anthrax. So he experimented with Bacillus anthracis, which is a rot, a gram-positive rot, that causes anthrax in animals and humans. And after he did these experiments, he was the first one to prove that a particular microorganism can cause a disease. So this is known as etiology, basically finding the cause of the disease. We're not going to study this for now. There is a chapter where we have more specific details about these experiments that he did, but it was very elegant, the set of experiments that he set, and in a way that still is helpful for today, those postulates. And then the role of immunity in preventing disease, it comes from the fact that knowing that if someone has a strong immune system, the person will have less likelihood to have infections. And that helped for developing vaccines, so that the person indirectly will be developing a strong immune system. And then there was this physician, Lister, a surgeon specifically, that he started thinking about the relationship between microorganisms and disease. So he started to think about the experiments from Koch, from Louis Pasteur, and he applied it for surgeries. He started to use carbolic acid or phenol to treat the wounds of patients. And the mortality after surgery dropped. And then there was a nice observation by Edward Jenner to develop vaccines. So we will see a little bit over that. So then Pasteur showed that microbes under anaerobic conditions, basically due to the lack of oxygen, they undergo a process that is known as fermentation, in which sugars can be converted into alcohol. And then he also proved that microorganisms can spoil food and beverages, and he developed pasteurization. The milk that you get from the fridge in the superstores, for the most part, they have been pasteurized. And we will see later on what types of pasteurization do we have. And then he was, with this pasteurization, helping the manufacturers of these products to save money, because many of these, they exported their wine to other places, and that time that it took for the transporting led to these contaminants found in the wine to spoil the wine and to become sour. So pasteurization, few words, is the application of high heat for a short period of time to kill harmful bacteria in beverages. Some of these products that we ingest nowadays can be ultra pasteurized, what we call ultra pasteurization. So these will be like the milk or the juices that they sell in these Tetra Brick or cardboard containers. They can stay long periods of time without spoiling in the shelf. So here are some milestones in the golden age of microbiology, including discovery of fermentation, pasteurization, aseptic surgery by Lister, Joseph Lister. So if you have heard about Listerine, either the little films that you put in your teeth to help with bad breath, or that liquid that you use after you brush your teeth, well, it comes from the word Lister, the last name of Joseph Lister. And Koch developed germ theory of disease, in which basically you have this disease caused by microorganism. And he basically got a Nobel out of his discoveries, including making pure cultures. And then there was this researcher, this bacteriologist, Neisser, who discovered this bacteria, Neisseria gonorrhea, that causes gonorrhea, a sexually transmitted disease. And Finlay, which was the one who discovered the agent for yellow fever. So part of this germ theory of disease came from the fact that also in the silk industry, there were a lot of loss of silk because there was this silkworm, the disease that it was caused by a fungus. And in 1835, Agostino Bassi, he discovered the fungus that was affecting this. And also, Pasteur in 1865 discovered that silkworm disease is caused more than by a fungus, by a protozoan. And this Hungarian physician, Ignaz Semmelweis, basically helped to decrease puerperal fever. What is puerperal fever? Well, it's the death of females after having their babies because they had an infection in their vagina and then in their uterus that went into the bloodstream. and that infection can be caused by any microorganism. In the 1800s, people didn't wash their hands, and they didn't even have gloves back then, guys, to have deliveries of babies. Many people, they didn't wash their hands. They had deliveries of babies, and in order to take out the placenta from the female uterus, they introduced the hand and part of the wrist, I guess. And with the microorganisms that they have in the nails, in their hands, they infected the female bloodstream. They contaminated the bloodstream. So they developed fever, and more than likely they were about to die. Why? Because there weren't any antibiotics back then. So then Ignaz Semmelweis, he advocated for hand washing, preventing the transmission of puerperal fever. And then in 1860s, Lister, again, he used phenol to prevent surgical wound infections. 1876, Robert Koch discovered the cause of anthrax and developed these experiments where you have specific steps to follow to prove the cause of a disease into what we call the Koch or Koch postulates. I don't know how that is pronounced. And then for the other discoveries that we have for the golden age of microbiology. So Koch as well, he discovered Mycobacterium tuberculosis, which is a bacteria that causes lung tuberculosis, among others. Hess was given credit for adding a solid agent, or well, an agent that solidifies media to create an agar. And then we have the Petri plates, which are containers, plastic containers or glass containers that are round, that have a lid that holds this agar. It was developed by Petri, so that's why we call them Petri plates. And then the theory of immunity was developed by Ehrlich, Theodore Escherichs. Sorry, by Ehrlich, not Escherichs. I was thinking the one who discovered E. coli. So in the late 1700s, almost 1800s, an English physician, Edward Jenner, he developed the first vaccine that we know against cowpox. So that helped for people to become immune to smallpox. Smallpox is a virus, a DNA virus, that causes infections not only in the skin, but when it goes to the bloodstream, can go and infect the brain, causing encephalitis and then death. So he was working in a rural area where there were a bunch of milkmaids, and many people had smallpox infections. And those smallpox infections in the skin, it develops scars, and severe scars. Now, who cares about scars in the skin? It doesn't affect anyone so much for functioning, right? However, when this smallpox virus goes into the bloodstream and infects the brain, then the person can die. So the mortality rate for having smallpox infections was high. And he observed that milkmaids, either they didn't get the smallpox infections, but if they had it, it was a mild infection. And he was wondering why. Well, it turns out that the cows, they have a virus that is called cowpox that is similar to smallpox. They are cousins basically. They're from the same family. And then when these milkmaids were basically milking the cows, they had their fingers, the skin of their fingers, they were dry, they had little openings in the skin, and through this opening, the viruses from some cows, they went through these cracks in the skin into the skin of course, of the milkmaids. And that made them to develop antibodies against smallpox. So what he did is then to scrape a little bit the udders of the cow, and then that little powder that he got, he hydrated it, make a little solution. From that solution, he made a wound in the skin, in the shoulder of a little kid, and then he put that solution in there. And guess what? That kid didn't develop the infection. So he developed immunity against this smallpox virus. So then, because the original specimen that helped to prevent this infection to happen was a cow, and vaca is the Latin for cow, then this process of making this immunity through this solution was called vaccination. So it was a nice observation then. So, summarize in your own words the germ theory of disease. Not everyone at the same time, please. Take turns.
If you wash your hands, you don't spread the germs.
Okay, if you wash your hands, you don't spread the germs. That's very good. Now, what is the importance of Koch postulates?
Provided a standard for testing for diseases.
Mm-hmm. Thank you. What is the significance of Jenner's discovery? Yeah, he set the precedent, right, for vaccination. Very respectfully, of course, we have people who don't like vaccines or they don't believe in their efficacy. Very respectful. From my perspective, seeing that, okay, people can do whatever they want with their body. And at least I like vaccines. I believe in them. I mean, vaccines is basically the artificial way to get the infection without getting the consequences of the infection. And it is proven to be protective. Okay, now the second golden age of microbiology focuses in treating diseases caused by microorganisms because, I mean, the first golden era already proved that microorganisms exist and how you can control them in a way. The second then was, okay, how do you treat infectious diseases? So then that set the precedent to treat disease with chemicals, and this is known as chemotherapy. One of the first agents that was developed, it was called salvarsan, and this was developed to treat sexual transmitted diseases, including one of spirobacteria that causes syphilis. And this is an arsenic-based drug that it was kind of having its toxicity. So sometimes these drugs that they developed earlier, you will find out how they had certain toxicity. Now, specifically, some drugs that treat infectious diseases are called antibiotics. Anti because it's against bios, which means life. So these are naturally produced antibiotics that kill or inhibit other microbes. And some of them, they are coming from sources like bacteria or fungi. So we will see in the end of this semester which specific microorganisms produce these natural antibiotics. And we have semi-synthetic that were developed because some of these antibiotics weren't as efficacious as they supposed to. So one of the first researchers that started to think about these antibiotics was Paul Ehrlich. So he was actually a medical student when he started to wonder, okay, can there be a drug that only affects the microbes but not the person itself? So he coined that to be the magic bullet. So in 1910, he developed this salvarsan, the arsenic-based drug to treat syphilis. And then 20 years later, sulfate drugs, sulfonamides, were synthesized. So those were the two early antibiotics. And they were only specific against bacteria. The syphilis bacteria is called Treponema pallidum. It's a spiral-like bacteria. We'll see that later. And then in 1928, Alexander Fleming, a Scottish bacteriologist, he discovered the first antibiotic. They put in your textbook by accident, but just as Jenner, guys, most of the things come from someone observing carefully what they see in their surroundings or in their experiments in this case. So Fleming, he was working with the Staph aureus, which is a bacteria, it's a gram-positive bacteria. And then he had several pretty plates with these bacteria, and he already knew what these colonies look like, the size and the color and the shape, of course. But he started to observe in some of his petri plates that some of these colonies, which are tiny groups of bacteria that grows in the surface of this agar in the petri plate, they were smaller than the usual ones. And then he started wondering why. And it turns out that every time that he saw these smaller colonies than the normal size, he always saw a big fuzzy colony around these small colonies. And then he found out that actually that big colony was inhibiting the growth of the bacteria, and that big colony, it was made by a fungus, a mold, Penicillium chrysanthemum. So this Penicillium chrysanthemum basically released this chemical, penicillin, that inhibited the growth of this bacteria. So then in the 1940s, penicillin started being produced massively, and then it was tested clinically. And it was kind of good because it was during the early years, of course, before the Second World War started, but the Second World War ended, of course, there were many soldiers coming home, and then, of course, many people know that sometimes when soldiers are outside, of course, they will have sexual interactions, unprotected back then, of course, and then they came home with infections like syphilis, gonorrhea, and then they were given shots of this penicillin. Turns out that it was very effective to treat these infections. So then, instead of just relying on sulfate drugs or salvarsan, now we had a third drug, which was penicillin. It was pretty effective. And then here, what you can see, it is this Petri plate. So this is a top view of a Petri plate. And all of these white spots that you see here are colonies of Staph aureus. And this is a black and white picture. But these colonies are typically yellow. And you see some of them in here that are very tiny. And then you see this big colony that, of course, it doesn't look like the big colonies of the fungus. Yes. So basically, he knew the difference between what it was Staph aureus and what it wasn't. Normal from different. And I cannot say abnormal because this is a normal penicillium colony. So then there is this natural competition in the microbial world, in the environment, guys, that is known as antibiosis. In order to gain nutrients, bacteria can secrete their own antibiotics and then inhibit the growth of the ones around. So then there are some problems with some antimicrobial chemicals. Some drugs can be toxic to humans, especially if these antimicrobials target intracellular bacteria or intracellular agents like viruses. So then as well, some of these can develop these microorganisms resistant to antimicrobial drugs. In the 1980s, we started to see that Staph aureus strains, they were not killed anymore by this penicillin drug, and then they start to use another drug that is called methicillin, and then methicillin wasn't effective anymore, and then they start to use another one that's called vancomycin, and then they come up with names for the different strains of Staph aureus after they're resistant to specific drugs. For instance, you might have heard about MRSA, which is basically the way to say MRSA, which stands for methicillin-resistant Staph aureus. And then we have another one that's called BRSA, vancomycin-resistant Staph aureus. For the ones that are the strains that are completely resistant, we have something that's called BZAP, or vancomycin intermediate resistant Staph aureus. So then, in the third golden age of microbiology, from the late 1980s to the present, the third golden age focused on overcoming problems of this nature, of resistance. Okay, so now to see your understanding. So what was Ehrlich's magic bullet?
It was
Or something that could destroy pathogens without harming the host.
Mm-hmm. You said it better than I. Thank you. Okay, so now we're going to see a little bit more of the history of microbiology. It's always interesting. We're going to define bacteriology, mycology, parasitology, all of these branches of microbiology. And we're going to see a little bit about the importance of microgenetics, molecular biology, and genomics. It's very simple. Anytime that you have in science, the ending logi, logos means study. And then that's the suffix, prefix tells you what you're studying. Bacteriology, study bacteria. Mycology studies fungi. Mycos is fungi. Parasitology studies both protozoans and parasites, because protozoans, for the most part, if they're affecting a living organism, it will be a parasite. So here we have a view of Guinea worm, which is called, well, properly, the name is Dracunculus medicensis. So this actually that you see here in this, I guess this is like a nail. This is actually the parasite. So it has been removed from the subcutaneous tissue of a patient, winding into a stick. And this procedure may have been used to basically for the design of this symbol, which is the symbol of the medical profession, a snake around this stick. So this Dracunculus medinensis, it enters into the skin of the patients, especially in the legs, and it can create these long extensions. That's like the name, that's actually the actual length of this parasite, and it can create a lot of problems. Now, immunology is the study of immunity. Includes basically developing vaccines and interferons, which are drugs that are used to prevent and treat viral diseases, including hepatitis C. And then a female researcher who helped to classify streptococci into different species based on the antigens found or the carbohydrates found in the cell wall components was Rebecca Lansfield. This classification, it is still used nowadays, and she made these nice studies that help us to know and understand this bacteria. So that specific antigen, it's a polysaccharide. And then she basically spent years and years developing this classification. Virology had two researchers that were critical for demonstrating the disease that was affecting tobacco plants. It was Dmitry Ivanovsky and Wendell Stanley in different times that discovered the cause of mosaic disease in tobacco. And it was in this discovery that electron microscope made possible to study these viruses in detail, to show the shape and also the size of these viruses. Now, for molecular genetics, we have microbial genetics specifically that study how microbes inherit traits. So you can have genetic transfer of information between microbes, genes passing along that can make them more aggressive. And microbial genetics is that specific field that study that. And then molecular biology basically study how genetic information is carried in molecules of DNA. And it can be from any species. So in the field of molecular genetics, we have some important discoveries like George Beadle and Edward Tatum. They show that genes encodes a cell's enzyme. In 1944, Oswald Avery, Colin MacLeod, and Maclean McCarty developed these experiments to show that DNA is a hereditary material. Like they show that because people, they believed before that proteins or even sometimes lipids, they were the genetic material, but they did some studies in bacteria where they showed actually that DNA is actually the hereditary material. And of course, who doesn't remember the discoveries of James Watson and Francis Crick that proposed a model for DNA structure? The name is not there, but Rosalind Franklin, the radiologist, she had nice pictures showing from the top the double helix. Somehow, sometimes not the more critical discoverer, from my perspective, get the honor that they deserve. And in 1961, Francois Jacob and Jacques Monod discovered the role of messenger RNA in protein synthesis for translation. And then we had later on the development of the field of genomics, which is basically the study of the entire genes in an organism. So with genomics, we have now new tools that can detect and classify microorganisms much rapidly than before. Before they use different techniques, which was very slow. And then this genomics has also enabled us to understand the interaction of microbes in their diverse niches in the different places where they are located in the environment. And also to understand if that interaction can lead to diseases, not only of humans, but also plants and animals. And now, one of these, I guess, it's my favorite researcher of the new era of microbiology was Paul Berg. In 1960, he developed recombinant DNA technology. How he did that? Well, he grabbed the insulin gene from humans and he inserted it into E. coli. He cut with restriction enzymes, these from DNA of humans, and then he cut a piece, he opened up the DNA of this E. coli, and then he combined this piece of the human gene with a piece of the E. coli. He ligated with ligase enzymes, and then he transfected these bacteria into cells that were capable of developing this insulin. Before, insulin was used to treat, of course, diabetes in patients, but it was extracted from pancreas from pigs. And the pancreas from pigs not only produced not so much insulin, generate as much insulin, but many people develop sensitivity because insulin from pigs is different from insulin from humans. So then they develop hypersensitivity reactions, and it was not good. So then because of this transfection of these bacteria with this human gene, we develop insulin in great amounts. We purify this insulin, and nowadays that's what we use through this recombinant DNA technology. And this is the summary of this first golden age, the second golden age. And I'm not going to go into each of these branches, rather I'm going to ask you differentiate microbial genetics from molecular biology and genomics.
Microbial genetics is studying genetic inheritance of microbes,
Mm-hmm.
and molecular biology is studying the genetic transfer of information and how that translates to proteins in all organisms.
Mm-hmm.
And genomics is just the study of genes.
Very good. And then this genomics, it can be used anywhere. Okay. So it's very important. So now microbes and human welfare. So we're going to list at least 4 beneficial activities of microorganisms. We're going to name 2 examples of biotechnology that uses recombinant DNA technology and 2 examples that do not. So beneficial uses of microorganisms to recycle materials, bioelements. So microbial ecology basically studies the relationship between microorganisms and the environment. We have specific microorganisms in the environment, and they can use specific nutrients. If we have an environment that has sulfur-rich areas, microorganisms that utilize this sulfur will be found in there. So they adapt and they use whatever they have available. Bacteria can use not only sulfur, but also carbon, oxygen, nitrogen, and phosphorus. And all of these elements are found in DNA, they are found in proteins, and they can get it from decayed matter, recycle it so that plants and animals benefit. So if an animal dies in the wilderness, there is no one to take care of this animal there. There will be bacteria in the environment that they will come and decompose the different components or structures of this animal, and ultimately these will be assimilated by the soil, and then if you have plants around, of course, these plants will grab all these elements, they will make their own structures, organelles, etc., and then other animals that are living will go and eat these plants, and then you start this cycle. So it's important then to understand that bacteria, if it weren't for bacteria, then we didn't have this recycling process. And then in sewage treatment plants, we have microbes that can degrade solids to clear the water so that the water can go much cleaner into a river or into wherever this water is released. So we have bacteria specifically, for instance, that they can degrade cellulose with cellulase enzyme. And other ones, they can use all of these organic material that can be found in the solids. And not only these can be utilized, but also can be released as a byproduct. So we have bacteria, for instance, that can use carbon dioxide as a substrate for energy. And then some of these bacteria can be used in bioremediation. When there is a spill of oil in the middle of the ocean, uh, bioengineers. They come and they bring these bacteria, and these bacteria use this oil and start degrading it so that it won't affect the environment. Mercury spills as well can be degraded by bacteria, organic matter in sewage. So here we have composting in a municipal waste plant. Yes, it emits gases that sometimes if you live around that area, you can smell it, but it's part of the process of bioremediation. Some microorganisms can be used to control pests from crops. So microbes can be only pathogenic to the insects. So they can be used as an alternative for chemical pesticides. And then one of these examples is Bacillus thuringiensis. So this can infect only the insects, but doesn't affect the plant or the animals. And some of these bacteria basically have toxic genes that can be inserted into some plants to confer insect resistance. And they can also produce crystals of proteins that is basically toxic for the insects. So here we have a picture of Bacillus thuringiensis. So this is a gram-positive bacteria. So this is a rod, this is the toxin that it produces, and this is an endospore. This is called parasporal body. It's toxic to insects as they ingest this bacteria. So biotechnology uses microbes for practical applications. They can produce food, they can produce chemicals. I was listening this morning to the news on NPR, and I find a kind of a nice use of some microorganisms. If you like chocolate, and you know that the prices are going high, well, you'll be happy to know that they have been working with some microbes that actually can produce flavors for chocolate, guys. So they can have basically nowadays a different substrate that has a texture as chocolate, and with this bacteria can produce the chocolate flavor. Can be artificial. Hopefully they will start doing now that for coffee, because coffee prices are also going up a lot for those of you who like coffee. And then recombinant DNA technology basically is more specific. So this is broad use of microbes, but this one is enabling bacteria and fungi to produce a variety of proteins, vaccines, and enzymes. So this one works with bacteria and fungi. And also it is used to, well, one of the uses is actually to help to replace or to add missing genes and replace defective genes. This is called gene therapy. Has been used to help people, for instance, with sickle cell anemia. And then some agricultural applications is that genetically modified bacteria that is used to protect crops from insects or from freezing GMOs. Something that I forgot to tell you the other day was about bananas, how they ripe. So you may have seen when you get your bananas, they are still green or yellowish. And you see like a little dark, well, brown spot, tiny one, and you start seeing a lot of them, that they are tiny, they are semi-oval or oval. Well, it's actually a fungus that lands there, and that helps to ferment the sugars from the bananas. So that's good. Like if you can grab a banana, you can wash it to wash away any fungus, and then put it in a Ziploc bag, seal it. It will never ripen. It will stay green, and it will not taste well. Okay. So sometimes you want some things there, but sometimes you don't want them. Okay, so 2 beneficial uses of bacteria. Name 2 beneficial uses.
Composting waste and sewage treatment.
Mm-hmm. Next, differentiate biotechnology from recombinant DNA technology.
Biotechnology is the use of microbes for their practical applications, but then recombinant DNA technology is basically taking that a step further and actually enabling the bacteria and fungi to produce different things such as proteins, vaccines, and such.
Mm-hmm. Yes, so recombinant DNA technology is more specific, and it is a branch of biotechnology. So now we're going to see what is resistance, biofilm, and emerging infectious diseases. So if you remember, we said that we have more bacteria in our body that is non-pathogenic, is part of the normal microbiota than normal cells. So then, those microorganisms that are present in and on the human body are called normal microbiota, and they prevent the growth of pathogens by occupying receptors that the pathogens may use to enter into our body. And then microbiota in our intestines, they produce vitamin B and vitamin K. And resistance is the ability of the body to ward off disease. So that's immunity, part of immunity. And then we have 2 types of immunity. We have innate and we have adaptive. Innate will be physical and chemical barriers that prevents the disease to happen. That includes the skin, mucosas, and its secretions like stomach acid, the tears that we have in our body, urine, sweat, sebum, the acid mantle that we have in the skin, and including proteins like complement system that exist inside our body, and cells like natural killer cells, which are part of the lymphocytes. And then we have part of the adaptive immunity, which includes lymphocytes T and B, that once we're born and we start getting in contact with several microorganisms and other things in their environment, they get trained to respond or not to respond to antigens, which are basically molecules that exist in our cells or they are part of the items that we can encounter in microbes and in the environment in general. And antigens are molecules that can elicit an immune response so that we can gain immunity. Biofilms basically are groups of bacteria that lives in specific areas, and typically they form layers. So this is a way to protect themselves. So we can have biofilms on rocks, pipes, teeth, medical implants, and basically some of them, they can be beneficial because they can protect the mucous membranes from harmful organisms, and they can provide food in the aquatic environments. Others, they can be harmful because they can clog pipes, cause infections, and make resistance to antibiotics. Okay, so we will leave it up to here, and I will see you tomorrow